All right, so we have been talking about Mendelian genetics. So that's like the basic genetic stuff that you probably learned in middle school and in regular biology. And it's basically just like, hey, you have two copies of every chromosome. You're a diploid organism, right? And basically you have an allele on each of those chromosomes. And so that's going to determine your genotype, which determines your phenotype. That's kind of like ultimately what you were supposed to get from the previous chapter. But now as we move into this chapter, I find it useful to kind of take a step back and look at those traits in a different form. So this here is a pedigree. Okay, so this is called a pedigree. And basically on any given pedigree, you can track a single trait. And that trait can be anything. So I'm just going to put trait up here. Anytime you have a pedigree, you want to tell the person looking at it what the trait is. So you will need to be able to create these pedigrees, but for now, I just want you to be able to like analyze them. So every pedigree has only one trait being tracked. So perhaps the trait is red hair. Okay. So if that is the trait I am tracking, what that means is that any individual who is shaded in has red hair. So this person right here has red hair. This person has red hair. This person has red hair. And these two gentlemen down at the bottom have red hair. Okay. So basically, a pedigree is a family tree. Okay. So these could be these individuals all have names. Ideally, you would have a pedigree with a data set that you could name everyone. So like, oh, here's John and here's Jane, and they had 4 kids, one of which had red hair, blah, 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 blah. Okay, we're going to practice with this. But just realize that these are all people. And anytime you have mating happening, we show that with this horizontal line connecting the two. And then off of that horizontal line, we draw all of their children. Okay, so those are the 4 children there. Anytime you have a circle, that is a female. Okay, so circle represents a female, and the square represents a male. Now, in this particular case, on this pedigree, you are tracking red hair. Okay? Traits on a pedigree can be anything, whatever it is that you're interested in. And so this one I just came up with arbitrarily, but it might be a disease, or it might be some kind of a genetic mutation that actually helps somebody's fitness, etc. It could be a really good thing, it could be a really bad thing, or it could be a neutral thing. So in this case, we're looking at red hair, and I just arbitrarily made this up. This is not a real family that doesn't have the real trait of red hair in it, etc. But just remember as we look through this that all the circles are females, all the squares are males. And anytime you see this slash through the individual, that means that they are dead, they are deceased. Okay. And sometimes that will be relevant. Sometimes it will be that they died due to the trait, like if it's a disease. And sometimes it won't be relevant. So, like here, you'll notice that, the older ones are more likely to be dead. And that's not that surprising because, like, you have 4 generations here. Not a ton of people have their great grandparents still around, right? It does happen, but it's not super, super common. So as you go up the pedigree, you might find that you're more likely to have deceased individuals, dead individuals. But what a pedigree really is it's a visual representation of information, right? Like you can think about your family right now and you know little things about all of them. But if you were to pick a trait, like you could say, if your trait was brown eyes, right now I want you to think about everyone in your family who has brown eyes. Okay, now tell me how many people in your generation have brown eyes. That's really, really hard to do if you don't create a visual. Okay, so that's what a pedigree is. It's just a visual. And so here I can basically say, okay, so I see 5 individuals who all have red hair. And there's some other patterns I could notice. I could say, oh, some of the individuals have red hair, but their parents didn't have red hair, right? So I can look over here, like, oh, okay, this individual right here has red hair, but if I look at his parents, no red hair, okay? So maybe that means it's recessive. We need to start thinking about stuff like that. I might also notice that all of the affected individuals are males. There are no females with red hair in this case. Okay? So that might be relevant. Maybe it's on an X chromosome, and we'll talk about that stuff. But a couple other things I wanted to talk about just in general for pedigrees is sometimes, depending on the trait, we won't include a parent's spouse. Okay, so like looking right here at this individual in the third generation down, she just has her children going right off of her. And that's totally fine. Sometimes pedigrees will show you a mate, like they will show a mate over here, and then it'll lead down to all the children. Sometimes it won't. If you're ever not shown a person, clearly there was a mate. This mother could not just have have 3 children by herself. Okay? But it could be that this pedigree isn't concerned with that individual because they know that he didn't have the trait or even the allele. Or it could be that that individual is not part of the family. And so you're working with limited data. And so we're going to practice some of those limitations throughout class. But really, I need you to be comfortable with looking at pedigrees and trying to figure out what they might be telling you. And pedigrees are great because it helps us to recognize kind of what plays by the normal rules and what plays by its own rules. So over here, we saw something that seems to play by its own rules. I see that only males are affected. And that's kind of weird. When we were looking at Mendel's pea plants, flower color didn't affect males and females differently, right? Pea pod shape or pea pod color or any of those things weren't affected by male or female differences, okay? So there are some exceptions to Mendel's rules, and because they are exceptions, we call them non-Mendelian exceptions or patterns. Okay, so this is something that doesn't really get explained by Mendel's ideas. Mendel has a lot of great ideas, and they work for a lot of really, really simple situations. But turns out that organisms, especially an organism as complicated as a human, are extremely complex. They have tons of traits. Their traits are determined by lots of different genes. It's not just a single gene all the time. And so sometimes we need to expand our understanding, expand our model in order to accommodate that. So there are a few different non-Mendelian inheritance patterns I want you to be familiar with. So one of them is polygenic. So poly means many, genic means genes. So some traits are determined by multiple genes, right? So like your height, there's not a single height gene. Like there's not just one gene on chromosome 4 that says, okay, you're either tall or short. There's a lot of genes that go into height. Okay, so that would be a polygenic trait. Height would be a polygenic trait. Sex linkage would be any trait that is on the X or Y chromosomes in humans. Okay, so it's on a sex chromosome. Other organisms have different sex chromosomes, but generally we're going to be looking at X and Ys because that's what humans have. Now, we're going to talk briefly about non-nuclear inheritance, but first I want to show you this codominance. Codominance is basically when 2 alleles. Remember how when you had alleles, you only showed one of them. So like if you were big P, little p, that meant that you had purple flowers because purple was dominant to white. You remember that from last lecture? Well, if a trait is codominant, it's actually a little different because then, let's say that you have 2 alleles. Let's say you have R and you have little r. Okay, so big R and little r. Big R stands for red, and little r stands for white. Okay, and in this case, so now we're not looking at normal dominant recessive stuff. We're looking at codominance. So co means together, and dominance means that you dominate over the other one. So there's 2 things that are dominating together. So in this case, if you are an RR, what flower color will you be? Well, I'm a red, so I'm going to be red. If you're a little r, what flower color? Well, I'm white, so I'm going to be white. But if I'm a heterozygote, a big R, little r, and it's codominant, I'm actually going to show both. I'm going to show red, and I'm going to show white. So that's what I have going on in this picture right here. Okay? It's not that I'm blending them together to make a pink all the way around. I have red and I have white showing. Both alleles are being expressed. So that's another thing that doesn't really play by the rules. And I know that this is a lot to throw at you all at once, and that's why we're going to practice each of these different examples of non-Mendelian genetics, but I need you to start thinking about them. So one classic example of a polygenic trait, so again, this is a trait that is affected by lots of genes, not just one, is eye color. So in middle school, maybe you did, a really basic little activity where you made babies with another student and you figured out their eye color of the child or whatever it is, right? And a lot of classes do that. But in reality, it's much more complicated than that. Eye color is not a single gene. There's lots of genes involved, and that's why we see lots of different eye colors, right? All of these are real eyes that you can see in the real world. And so it makes it difficult to predict what eye color your child will have in reality because it's not just one gene. You can't just do a simple, easy Punnett square. If you're going to try and do a Punnett square, it's going to be a big one. So a lot of different traits fall into this category of having multiple genes associated with them. Skin color is another. If there was only one gene for skin color, you'd either have dark skin or light skin. But that's just not the case. We have a lot of different pigments of skin, okay, from extremely, extremely pale to extremely, extremely dark. And that is a result of many different genes having many different alleles. So human skin color specifically has at least 3 genes, and that means that there are 6 alleles because each of those has at least 2 alleles causing skin color. And so that's why we see a really big normal curve of skin color in the population. Okay. Now, when we look at large data sets, you're going to see over and over and over again this normal curve shape. Just remember that a normal curve is going to be more likely if we look at a bigger data set. So if we have more people that we're sampling, we're going to see more normal curves. So just as a friendly little reminder. Now, I mentioned this earlier, but some traits are what we call sex-linked. And that means that they're on a sex chromosome. So it could be on the X chromosome or it could be on the Y chromosome. Okay? So generally, the Y chromosome, like in humans and a lot of other animals that have the Y chromosome, it's very small and there's not a lot of genes. So generally, if you're looking at a sex-linked trait, it's going to be dealing with the X chromosome. Remember that this individual on the left is a female because she has 2 X chromosomes. This individual on the right would be a male because they have 1 X and 1 Y. So if I were to look at this first X chromosome, this little area right here, that's a gene. Okay, and that gene says color. Okay, that gene codes for proteins that let you see color. Okay, and then right here, the same spot has a color gene, and right here, the same spot has a color gene. Okay, but the Y chromosome doesn't have that spot. Okay, it does not have the same gene, so there's no color gene on that Y chromosome. So that becomes a little complicated because males only have 1 X chromosome. So that means that they only have 1 allele, right? We keep talking about how every individual is has 2 copies of every chromosome. They're like, oh, you're a big R, you're a big R, or you're a little r, right? We kept seeing that. But now we're talking about a situation where, well, you might only have 1. So we need to come up with an annotation for this. So like if this individual, this female individual, was a carrier for color blindness, okay, that means they're going to have 1 normal allele. So I'm going to give it a dominant capital C and 1 recessive allele, little c. Okay. And this individual over here, this male individual, has a little c, and then they don't have another X chromosome, so that's the only allele they have. So if I wanted to write the genotypes of these individuals, the one on the left is going to be, and I'll write it over here so you can see it a little better, is going to be X big C, X little c. That's their genotype. Okay? Their phenotype is normal. They can see all the colors. Okay? This individual, this male individual on the right, has a little c and then a Y chromosome, which doesn't have any C because it doesn't even have that gene. So this individual is actually going to be colorblind. Okay? They only have 1 allele, and in this case, that allele is the recessive trait that says, hey, you're colorblind. Okay. And so what you're going to notice is that a lot of sex-linked traits or sex-linked diseases are going to be more common in males because typically females will have a dominant allele somewhere, right? In one of their two sets, one of them is going to be dominant. But males only get one. So if the one they get is recessive, they're going to have that trait. We're going to explore some examples of that. This is just a little reminder that these are all just probabilities. Okay, so on the top here, I have the father and the carrier mother. So the father does not have whatever trait it is that you're tracking. The mother has one copy of the allele. So what I want you to do is kind of just think about this as being a recessive disease. Okay? If I have an unaffected father, and that means they don't have it, then I'm going to give them a capital letter. It's dominant, right? So I'm going to just do big R. Let's just be, consistent. Okay, so that means that all of these dark boxes like dad had right here are going to be X big R, and all the white boxes are going to be X little r. So mom can pass either X little r or X big R. Dad can pass either X big R or Y. And so here are all the different possible children that they could have. They could have an unaffected son. Okay. They could have an unaffected daughter. They could have a carrier daughter, or they could have an affected son. So if mom passes X little r, so that's the affected allele, to the son, and dad passes the Y chromosome, thus making it a son, then you're going to have a son with that trait, whatever that trait is. Maybe it's colorblindness. Okay. If you have mom pass the X big R and dad pass the X big R, then you're going to get this child right here. Two normal alleles. That's why they're both colored in on the chromosomes. If dad passes the X big R, but mom passes that diseased allele or that recessive allele right there, well, then we're going to make this child because it's an X big R and an X little r. And if dad passes the Y chromosome, but mom passes the X big R chromosome, you're going to get an unaffected son. It's all just probability. You could have, instead of drawing a diagram like this, drawn a Punnett square just to represent the different possibilities. So I could put mom on top. Well, she could pass an X big R, or she could pass an X little r. Dad could pass an X big R or a Y. And so you just fill in all of the possibilities. So like this individual would be an X little r Y, so they're right here. This third individual over is the carrier daughter. So that means that she has the trait, she has the allele, but she does not show it. So she doesn't have the disease, if it's a disease. So she's right here. Okay. This is an unaffected daughter. So she got two normal alleles from her parents, both on the X chromosome. And here's an unaffected son. Got the normal allele from mom and the Y chromosome from dad. So these are just showing the same thing. Okay. Now, a lot of these traits are pretty minor. So you'll see stuff like milk production in females, or you'll see baldness patterns in males. But there are some really serious diseases that are sex-linked, like hemophilia. So we're going to be exploring some of that in class. But hemophilia, just to kind of prime you on that, is basically a disease that prevents your blood from being able to clot. Okay? And when you think of blood clotting, you might think like, oh, that sounds bad. That sounds like a stroke. But that's not always bad. Sometimes you want your blood to clot. Anytime you get a cut in your blood vessel, you want it to close. And in order to close, it needs to kind of seal. And that's what clotting does for you. It lets you seal that hole. And so if you can't clot, you're just going to keep bleeding. And so that's what individuals with hemophilia have. So we're going to explore some of that later. But typically, if you don't do anything to help these individuals, they have a life expectancy of about 10 years because they will get a paper cut and bleed, or they will fall and get, a big scrape and they'll bleed. There are a lot of different treatments nowadays, but now we will typically see infusing factor VIII, you don't even know what that is or anything, from individuals who can make it, so just a normal individual, and introducing it into the individual's bloodstream. So it's basically just a nice little relatively easy treatment that we didn't have that long, very long ago. It's relatively recent. So Punnett squares can be used for sex-linked traits just like any other trait. And if we were to do this for hemophilia, we would have to see like, okay, what does X big H mean? What is Y? What is X little h? What is X little h? Okay, so in this case, X big H is normal. Okay. Y chromosome doesn't have the gene, so we still have to write it because you have a dad who has a Y chromosome, but it doesn't have the gene, so there's no H. There's no big H or little h there. Mom over here on the left can pass either an X little h or an X little h. Okay. And this is all a hypothetical situation if the female, the mom, has hemophilia. So she has hemophilia because she has an X little h and an X little h. It's recessive, so you need to have both X little h's, but the normal dad has just an X big H. And so these are the possible offspring. You could have this individual, which has two X chromosomes, so that would make it a daughter. And they have a dominant H, so a regular normal allele, and a recessive allele. So this individual would be a carrier. Okay. This individual down here is also a carrier. This individual has an X little h Y. Okay. So that would be a male because it has a Y, and the only H they have is a little h, so they would actually be hemophiliac, and you would get the same thing here with the X little hy on top. So all of their sons are going to be hemophiliac, and all of their daughters are going to be carriers. So these individuals right here, the daughters are not hemophiliac, but they do have the allele. They're carriers. So like if they went on to have kids of their own, they might pass that on to their kids, and then you might get kids that have it, etc. So here's another example. I recommend that you take a look at it, you pause it, and kind of analyze what's going on in this Punnett Square, but I'm not really going to spend any more lecture time on it. And this kind of takes me to non-nuclear inheritance, and this is something that I just want to just barely brush on, because there are some organelles in the cell that kind of play by their own rules, right? So mitochondrial DNA is a thing that you might have heard before. Basically, you get your mitochondria from your mother, and mitochondria has its own special DNA. And so if you're tracking a trait that happens to be on their DNA, it doesn't follow the same rules because you don't look at dad's DNA. And I don't need you to memorize that or anything. Just know that there are other cases that are unique. And there's yet another thing that I would love to spend a ton of time talking about, but we just don't have the time. But just so you know, for all animals that we're going to be worried about, the mitochondrial DNA, so that's the DNA in the mitochondrion, is passed through the egg, but not through the sperm. Okay? So your mitochondrial DNA should match that of your biological mother's. Okay? And you should have none of your father's mitochondrial DNA in your cells. Okay? Here are some extra resources that I would recommend. This is a really beefy little section because it's basically saying like, okay, last chapter was interesting and useful, but sometimes it's not enough. And so we need to explore that. And really what you're going to need to do is practice, practice, practice. This is a unit where it's really important to make pedigrees. It's really important to track traits and try to figure out how they're inherited. If I showed you a pedigree, could you maybe figure out whether it's on the X chromosome or not? It's challenging, but it's something that you do need to be able to do. Okay. So with that, that's going to be the end of non-Mendelian inheritance. And I'll see you guys next time for DNA structure.